our study sort of had two parts. It really looked at what's happened with the FDA's accelerated approval program over the last 10 years. And as many people will know, the accelerated approval program, although it was initially brought about to treat HIV, it's now mostly used in oncology, with more than 80% of recent accelerated approvals occurring in the oncology space. And so it's of real interest to how drug development occurs in oncology and how clinical trials are designed in oncology. So we took a look at the last 10 years of the program and its use in oncology. First, we wanted to know, well, there have been some concerns raised in the past about how many oncology drugs that are approved with accelerated approval go on to show true benefit, true clinical benefit in clinical trials. And so we wanted to update those analyses and look at, well, has that improved as time has gone forward? And the second part of our analysis was to look at when those accelerated approval drugs are converted by the FDA from accelerated approval to full or regular approval, what is the basis for that conversion decision? And so we looked at, over the last 10 years, all of those conversions from accelerated to full approval. And we said, well, how is the FDA making those decisions? And so if we go to the first part of our study, really what we looked for was what proportion of these drug indication pairs go on to show a clinical benefit in their confirmatory trials. And we define that as a benefit in either overall survival or a benefit in quality of life, or both, of course. And what we found was that about 22% of these drugs went on to be withdrawn from the market because they didn't show a clinical benefit. About 15% of these drugs at more than 5 years were still ongoing accelerated approvals, which means that their confirmatory trials have yet to give us an answer about whether they show clinical benefit. And then we showed that the other approximately 60% had indeed been converted. But of those converted drugs, there was a proportion of those, nine of those that had actually not shown a benefit in either overall survival or quality of life. And so that meant that overall, actually a majority of accelerated approval drugs had not yet shown a benefit in a clinical outcome. And that's really for two reasons, partly delays and partly the design and endpoints used for many of the confirmatory trials. So I think most oncologists and our patients are generally going to be happy if we know what we're signing up for, if we know what we're getting into. So if patients are adequately informed about what we know about drugs' efficacy and toxicity, as you highlight, then patients can make informed decisions about whether they want to pursue drugs that have promising evidence but not full confirmatory evidence. Really what we're after here is not about which drugs are good drugs and bad drugs, but about certainty in clinical benefit. How can we ensure that even if there's a period of uncertainty when patients can choose and clinicians can choose to use drugs with some uncertainty, how do we resolve that uncertainty? And how do we resolve that uncertainty in a robust way and in a timely way? And really that is our focus, is on trying to resolve that uncertainty. Let me pick an example from the hematology area. Let me talk about bosutinib. There was a randomized controlled trial called the BEFORE trial that compared bosutinib with imatinib in patients with chronic myeloid leukemia. And really what that showed was that yes, there was an improvement in what we call the major molecular response, the number of patients whose CML became undetectable in the blood. However, there was also an increase at the same time in toxicity, particularly GI toxicity, but a number of different toxicities. And so then it becomes a very hard question, both for clinicians and for patients, which is, yes, this does have a higher chance of getting my CML into major molecular remission, but it also has a higher chance of causing me side effects. And so without good, robust, high quality clinical information, particularly patient-reported outcomes, particularly quality of life data, it becomes very hard to know which decision to make in that. That's one example, but this occurs quite frequently across different indications and with different drugs. And that's not to say that bosutinib is not a good drug, it's just to say that the way the trial was designed and reported leaves residual uncertainty about how that drug should best be used and when that drug should best be used. So really, it's about trying to resolve that uncertainty for patients and clinicians. I think it's really important that when clinicians are counseling patients about trying some of these novel therapies that have received accelerated approval, that they really ensure patients are well informed. For example, that many of these therapies have not yet been shown to extend or improve people's lives, and that patients and clinicians are very much entitled to try these therapies, and I think it's great in many cases to have early access to some of these therapies. For example, in hematology, I think the bispecific antibodies across lymphoma and myeloma are very promising drugs, but at the same time, there are some concerns around infections and toxicity. And so I think as long as patients and clinicians are having open and informed discussions about these promising novel therapies, then they can make informed decisions about when they want to try these, as I say, very promising but just not yet confirmed drugs.